introduction. I've been a longtime fan of this seminar series, so it's pretty exciting to present here myself. Today, I'm going to talk about a new strategy that we've developed in the Satley lab to identify sets of genes in non-model plants and how we've used this to find the missing genes in the biosynthetic pathway for Taxol. Here. Okay. So about a third of a century ago, Congress passed a law protecting a certain tree because it contained a certain molecule. And that molecule, Taxol, was a promising new cancer drug that would go on to treat millions of patients with ovarian, breast, and other cancers, and would actually be the best-selling small molecule on the drug for several years. But this protection was warranted because Taxol is found at such low abundance that you need about 100-year-old trees to get enough of this compound for a single patient. And there was actually a lot of threats of deforestation and concern about environmental damage just to get material for the clinical trials. And now, about a third of a century later, Taxol still remains one of our best tools to fight cancers, especially with a number of derivatives that we have from it. But we still source all of this chemical material from the tissue of yew trees or yew cultures. Now, if we knew the 24 or so enzymes in Taxol biosynthesis, we could access this drug, presumably much more affordably and sustainably through biomanufacturing and synthetic biology. But of course, this requires gene discovery of those enzymes, which is the major challenge of genetics and biochemistry that I'm going to be talking about today, and I know many of you work on as well. All right. I think Taxol highlights how hard pathway discovery is in plants quite well. It's such a valuable molecule, and there's been this history of some really stellar biochemists that have worked on this pathway for years. But we still had missed a lot of the pieces that would have enabled us to assemble this pathway. And furthermore, despite missing some of the enzymes that would allow us to stitch this pathway together, even those first couple of enzymes that we've known for now about 2 decades, we've struggled to get to work in heterologous systems. So this first oxidase, this T5 alpha hydroxylase, when we express it in plants or fungal heterologous hosts or bacterial systems, it mostly produces these side products, which has really hindered all these efforts to try to reconstitute this pathway and suggests that we still miss some sort of comprehensive understanding of how this chemistry works. All right, so why haven't we been able to figure this out? I think, one of the general challenges of trying to dissect plant metabolism is just its incredible branching complexity. Each yew tree species makes hundreds of molecules that are very similar to Taxol. And these related taxane metabolites are synthesized by this enormous army of enzymes. We are seeking just a minuscule subset of these that are specific for synthesizing the metabolite that we're trying to study. And here I want to draw just a really sharp contrast between how this type of biological discovery is done in different branches of the tree of life. In prokaryotes, we have tools that very specifically tell us which genes function together because genes of a single function are nearly always encoded in these tight genomic clusters, often even on the same mRNA. And while we have a little bit of clustering in plants, it's just not nearly consistent enough for us to be able to rely on in any sort of systematic way. And this difference has some really profound implications for our ability to understand and discover gene function, especially when we're working in non-model organisms like yew trees. So just for example here, just browsing an uncharacterized microbial genome, we can automatically, confidently resolve entire pathways, even if we don't really know what most of the genes do or what the products are. And when we have a new organism, we can just catalog those gene clusters and get an immediate snapshot of the types of chemistry encoded in that organism. And then finally, this type of approach has really enabled a lot of discovery of new biology and chemistry. For example, association between these repetitive RNAs and nucleases led us to CRISPR. And then since then, we've been able to sort of recycle that process and discover all kinds of other RNA-guided functions. And for the most part, none of these types of analyses are really common in plants. just because we don't have this consistent clustering. When we want to know gene-gene associations, most of us generally rely on other strategies, primarily gene co-expression, but this approach still really suffers from a lack of specificity and from false positives. And specificity is just really crucial when it comes to working with uh, plant genomes because they're so big. And so I think our hunt for the Taxol genes exemplifies this painfully well. The genome of Taxus has something like 45,000, 50,000 genes. And within this haystack, we're trying to find just 20 needles. So here, each color dot represents real enzyme candidates that we have to filter through. And even if that co-expression can break them down into a couple of clusters, that's really not enough specificity. We really need to narrow this down into these very small specific clusters if we're going to not have to test hundreds of candidate genes. So we and many others have tried to find the Taxol genes just using bulk RNA-seq, looking at transcriptomic data and looking at gene expression across several tissues, often with some different conditions, to try to find which enzymes are co-expressed with some of the known Taxol genes. And this has failed for Taxol and some of the similar projects in our lab, probably for two reasons. First, we often struggle to find conditions where our pathway is really highly expressed. And secondly, even if we do, we often find that there are hundreds of unrelated genes, a bunch of unrelated biochemistry that's also expressed in that same organ or same condition. And so we needed to sort of redesign and scale up this experimental strategy so that it could give us a data set with two ideal features. First, we needed our genes, especially the ones in the pathway we cared about, to actually be highly expressed in some subset of our data. And we also needed enough conditions to see differential expression of these hundreds of different biochemical processes so that we could differentiate them and dissect them apart. And we devised a way to do this by leveraging single nucleus RNA sequencing. And this is not only to capture the natural diversity of cell types, but really it's to enable us to test dozens to hundreds of perturbations in a single experiment. And we can do this because plant tissue sections can be kept in liquid media, can be kept alive in liquid media, as anyone who's tried to start a callus knows. And this has allowed us to take tissues of Taxus and treat them with large panels of chemical or biological perturbations, literally arraying these needle tissues or root sections across 96 well plates of conditions, and exposing them to things like hormones or other modulators of stress, like pathogens or their associated molecular patterns. And then also some of the metabolites relevant to our pathways that might tap into some of the regulation of expression. And then after this sort of perturbation, we can just pool all of these tissues and rapidly wash them and combine them into just one single cell RNA sequencing experiment. And by sampling cells from these diverse perturbations, we can profile cell states that are transient or challenging to observe or dependent on a specific perturbation that we didn't realize was important. Now, the question is, does this help our search for the Taxol genes? Does it provide more specificity for these gene-gene associations? And we don't have much sort of gold standard gene sets in the U genome, but we can use the Taxol genes as a test case. So we can take this practical metric where we'll just rank all 50,000 genes in our genome based on correlation to, say, the first gene in the Taxol pathway. And then we look where some of the other known genes rank in that list. And if we do this with about 80 high-quality RNA sequencing data sets, samples, it works well for some of the genes, but some of the other genes in our pathway are still ranked thousands of genes down this candidate list, highlighting this problem of specificity, right? And then doing this with this new data really improved that ranking for some of these known genes. So this was our first indication that our approach was definitely improving our co-expression analysis. Okay, so we had this new data. We wanted to see if we could find a co-expressed gene module with these Taxol enzymes, including some of the genes that we knew and some of the genes we had that hadn't yet been discovered. And we do this with a strategy called matrix factorization to process this expression data and identify modules of genes with coordinated expression. We can then examine these modules to see if any of them are enriched, are dominated by some of the known Taxol enzymes. And we found not one module, but actually three separate modules where some of the top module genes included those Taxol enzymes. Interestingly, these modules roughly align with the proposed order of chemical steps, suggesting that we have these separately regulated subsections of Taxol biosynthesis. Now, if we look at which cells express these, we see that these modules are expressed in different cell states. And if we look at the top scoring genes for each of these modules, I'm highlighting in green the previously known Taxol enzymes, which you can see really dominate these gene lists. And in orange, these little orange dots here, I'm giving you a sneak peek of some of the new genes that we've now found to contribute to this biochemical pathway. All right, so let's talk about what we found. Like many of the people in this community, we often prototype our gene sets by expressing them heterologously in tobacco, in Nicotiana benthamiana, and then running LCMS. I mentioned at the beginning of the talk that the first couple of steps were really showing a lot of issues with reconstitution. So we can express our first terpene synthase and it works fine. But as soon as we add that second gene, this oxidase, we mostly get these highly rearranged side products, not the hydroxylated product the hydroxylated intermediate that we desire. And so we can take a look at the actual data for that. When we just start building our pathway stepwise with the terpene synthase, we get a very large amount of our first terpene intermediate. When we add that second gene in the pathway, we get a very small amount of the correct product here in red. And mostly in blue and green, we get these side products. So we wondered if maybe these gene modules that we had identified might illuminate some missing crucial component for this chemistry. And by going down this list and testing some of the unknown genes here, we found that the 13th gene in this list, when added to the pathway, dramatically improves the efficiency of producing the correct intermediate. Now, initially we didn't know what this is doing, so we just gave it this sort of agnostic name of facilitator of taxane oxidation, or PHOTO. So I'll be talking a lot about PHOTO for the next couple of slides. And when we add PHOTO to the pathway, it improves the production of our hydroxylated intermediate here by about an order of magnitude, and it completely eliminates the production of these side products that we were producing. So what kind of protein is this? We were really surprised that this gene had such a major impact on biosynthesis because it comes from a family of proteins, these nuclear transport factors that don't have any precedent in plant metabolism. The only plant homologs were relatively distantly related. The only characterized plant homologs were relatively distantly related, and they only had roles in protein scaffolding at the nucleus. This is just this small, cute little canonical alpha beta fold, and it has this C-terminal helix, which when we knock it out, we see is important for a function. And we were surprised to find that actually PHOTO's mechanism appears to depend on some direct interactions with the enzymes in the pathway. We found now binding with multiple Taxol enzymes, both in vitro and in vivo. And in vivo, this binding, which we can measure by co-IP, seems to be strengthened if we actually add our first terpene intermediate to the cell lysate, which is kind of an interesting observation. And furthermore, we were excited to see that there are actually a bunch of close homologs to PHOTO found throughout land plants. We have homologs that seem to be very structurally conserved in many of our model plants, including Arabidopsis and some of our major crops. You can see highlighted in blue here that conserved, that crucial C-terminal helix that we found to be functionally important. And none of these homologs had described functions yet. And in fact, when we look across land plants, we tend to see that most plants have just one PHOTO ortholog, suggesting that it's probably some sort of highly conserved and maybe even essential function. In gymnosperms like Taxus, there's been a couple of duplications of photo. So presumably photo has some conserved essential function paralog in Taxus, and then photo is probably some sort of neo-functionalized duplication um, in recent history. This finding was also very exciting because people have been trying to assemble this pathway in industrial manufacturing strains and have always been sort of stuck at this first oxidation product. And we were very excited to see that when we added photo to a yeast production strain with just the first two genes in the Taxol pathway, just like in plants, this photo can correct the chemistry um, in these yeast strains, enabling us to produce finally this second intermediate for Taxol biosynthesis quite cleanly instead of some of these side products. All right, so this was very exciting. We had solved this sort of major hurdle in just reconstituting this pathway, but we still had a bunch of missing enzymes that we had to discover in order to actually reconstitute the majority of the chemistry here. And to do this, we combined photo with 6 of the previously known enzymes in the pathway, express them in Nicotiana, and we could efficiently produce this early intermediate. And then we just went again down that rank list of candidates in our co-expression module. And doing so, we very quickly found this P450 oxidase. I was able to very efficiently convert this previous intermediate into the next by installing this 9-alpha hydroxylation up on the right here. As we move deeper into the pathway, we mined not just the first module, but the second and third modules for new enzymes. And just to sort of summarize a little bit of repetitive work that took a long time, we found a number of new enzymes in the middle of the pathway. And so this then allowed us to sort of build our pathway
Okay.
stepwise towards
I
Taxol.
think so.
Um, all right, build our pathways stepwise towards Taxol, decorating this molecule with these requisite oxidations and acetylations as we added these new enzymes. Beyond the requirement of that gene photo, we found some other surprises. The main one was just that our core model for the biosynthetic route seems to be partially incorrect. We thought that we would just have to add the modifications that we see in our final product, which is 9 oxygenations in red and 3 acetylations in blue, which I'm showing here on this on these X and Y axes for this biosynthetic route. That would be sufficient to give us Baccatin 3, which is just Taxol minus its side chain. However, as we were assembling this, we got to a point where we stopped being able to oxygenate, install the oxygenations that we needed. And by examining some of the metabolites that are naturally present in the tissues of yew trees, we realized that there were some compounds that look a lot like the intermediates that we were getting stuck at, but have some additional acetylations. And so this led us to hypothesize that maybe there are some temporary modifications, some acetylations that were required for the pathway to proceed through this stage of the pathway. We can look at the actual data for this. So we had built our pathway up to this stage here, this black molecule, and we had anticipated that it was the stage where we were going to incorporate one of Taxol's more unique chemical moieties, which is this 4-membered oxetane ring, using this recently discovered gene TOT. However, when we added TOT to our pathway, you can see that it has no effect on consuming this intermediate into a more oxygenated form. Now, previously in this discovery process, we had found this orange enzyme, this acyltransferase, that was able to install an acetylation at the C7 position up here. But because Taxol is not acetylated at the C7 position, I had sort of discarded this as unnecessary. However, if we feed TOT, if we add TOT to this pathway, we see that it's able to very efficiently install this 4-membered oxetane ring and move the pathway forward. All right, so it seems like kind of similar to synthetic chemists, our plant will protect and then deprotect some of the more reactive parts of this molecule. And indeed, when we leave free hydroxyl groups on our molecule and we work with it in benzamiana, we often see that they're converted into other things, especially ketones. And so maybe these acetylations are protecting these hydroxyls from some sort of similar reactivity in the cytoplasm of Taxus. Um, it looks like Taxus acetylates almost every oxygen, almost every hydroxyl that it installs when building Taxol, even those that are not acetylated in the final product. Of course, this meant that we ended up with an intermediate now that had these extra modifications, and we needed to go and find extra enzymes to remove them at the end of the pathway. But here, we could take that same co-expression approach. We tested some hydrolases from our Taxol modules and identified two that were efficient deacetylases capable of removing these temporary modifications at the end of the pathway. And these two deacetylases combined with one last oxygenase that installs this ketone at the C9 position allowed us to then synthesize this Taxol precursor baccatin in the leaves of Nicotiana with 17 enzymes from Taxus. And this was really exciting because modern industry actually harvests this molecule, baccatin, to produce the world's supply of Taxol and its derivatives. And we were now making it in tobacco at concentrations that were similar and maybe even a little higher than it's found in yew trees without doing any sort of metabolic engineering optimization. Um, so this is probably the most industrially relevant part of our findings, but we still were very curious if we could actually just produce Taxol completely and install that side chain. So theoretically, even before we started this project, the genes for that side chain were completely known. It initiates from phenylalanine, and when we added this gene set to our 17 gene set to baccatin, we saw no conversion. And we noticed that one of these genes specifically really didn't co-express with the rest of our pathway in our data set. This purple gene PCL. However, when we looked at this family of genes, which are acyl activating enzymes, we noticed that another member of this family, this gene PCL 867, very strongly co-expressed with several of the Taxol modules. So by swapping this new PCL in, we then were able to very efficiently convert our baccatin pathway into a pathway that produced this side chain functionalized form, which is dibenzoyl deoxytaxol. So now we were just 2 steps away from producing Taxol. While we were working on this, some other biochemists figured out some issues with those last couple of steps, and we were able to incorporate their findings now just recently to now produce Taxol from scratch in Nicotiana benthamiana. Now, at this point, we probably have the full enzyme set, but we're still finding other co-expressed genes that seem to improve yields, such as I'm just showing 2 transporters here that seem to really help with the Taxol production. So it seems like there's still more to this story to discover. All right, so just to bring all of this together, our method has allowed us to find 8 new genes in Taxol biosynthesis. Initially, we thought that we would only need on the order of 3 or 4, but several of our hypotheses at the beginning of this project were just wrong. The pathway proceeds through some intermediates that we didn't anticipate, and these temporary acetylations need enzymes both to add and remove them. We also needed some surprising genes like photo. Fortunately, our approach really prioritized genes with enough specificity that we didn't have to rely heavily on the assumptions of what gene was needed at each step along the way. And unlike conventional RNA-seq, this perturbed single-cell data really gave us very strong confidence that each of these new genes was involved. One of the more challenging and curious aspects of this project as we were assembling this pathway was how often we found genes with near redundant or redundant functions. I think this is a fairly common um, finding in plants, but in green here along the top, I'm showing 5 different regions of Taxol that have to be installed throughout the pathway. And for each of these chemical steps, we found at least 2 enzymes capable of doing this chemistry. So sometimes these enzymes show very little sequence similarity. One of these 2 P450s that in the set that installs this 9-alpha hydroxylation, we found 2 different P450s capable of doing this with only about 20% identity, very strongly suggesting that this is a case of convergent evolution. Now, these 2 genes have slightly different substrate preferences, and only one of them really worked well in our pathway. So we suspect that maybe the other is involved in synthesizing one of those other taxane molecules instead of Taxol. Um, for other sets of genes here, like the 2 options for our first step at the terpene synthesis, it seems like these 2 genes are functionally redundant as to the best of our experimental knowledge. Perhaps um, one of these genes is involved in synthesizing taxanes in different conditions or different tissues because they do have slightly different expression. But beyond that, we're not sure what the difference is here. All right. So in summary, we've developed this new transcriptomic strategy to identify gene sets, which provides a little more specificity for these gene-gene associations. We intentionally designed the strategy to be compatible with non-model organisms that lack any genetic tools. And we really hope that it would be useful for people in this community. I know many of you work with plants that are intractable or have large genomes. In yew trees, this method has allowed us to find several unexpected components of the Taxol pathway, which we can now use to produce um, de novo, both the industrially used precursor for Taxol and now Taxol itself. If you want to see more details, we did just recently publish this. We also have a manuscript with a little bit more information about photo coming out soon on bioRxiv. Um, there were a lot of people who helped with this project along the way, but I especially want to thank 2 talented grad students, Jack and Chloe, who both made some very significant contributions, and 2 precocious high school students, Alicia and Hannah, who spent summers working on plant chemistry in our lab. And I'm also very thankful for our funding sources, and thank you for your attention. I would love to take any questions.
Yeah, thank you so much for that wonderful presentation. For questions, please just raise your hands or feel free to put your questions in the chat. They're still thinking about questions. There's a question. Okay, great. Go ahead, Sam.
Sorry, I was actually I came in a bit late, I apologize, and I'm fascinated by this process, and I was just quickly switching to reading the paper and how you actually did all these perturbations and to explore and actually activate these biosynthetic genes. But the process it appeared it entirely Newton's transcriptomics, as I take it. And so, across, even though you're actually perturbating the plant tissue in so many ways and so many conditions, I take it that from any of those, you're not actually sampling the metabolomics at all. Looking for the abundance of any key metabolites. It's just all transcripts, right?
Yeah, that's a good question. So um, the question is about whether or not we are also measuring metabolites when we do this perturbation, right? Which, yeah, I think a strategy that I've tried before I did this approach, and several people in our lab have tried, is You try to find the tissue where the chemistry you're studying is happening. And if you can't find the tissue, then you try to find the conditions. And so one of the experiments I did before this pure transcriptomic approach was I just tried a bunch of these perturbations and I looked at metabolomics to try to see, can we see like increased accumulation of say labeled Baccatin or Taxol. And I couldn't find any conditions that would actually give me a real boost in those metabolites. Now that we know a little bit more about what the intermediates are, um, I was able to go back, look at some of those older data, and also redo this sort of perturbation now coupled to metabolomics, and get a better sense for which of the perturbations were helping to activate at least the first module of our biosynthetic process. But I think it's still important to point out that the scale of what we can test with the transcriptomic approach is just much bigger than what we can test with the metabolomic readout.
Oh,
Um,
absolutely.
and I think what we're really, the situation where the metabolomic readout doesn't help you is if you're really activating biosynthesis in a small set of cells, a small subset of cell types, then that signal might be just a little too small for you to see. But if you're doing single cell sequencing, then you can isolate those and see it.
Yeah, that's true. Okay, yeah, thank you. I guess it's followed, so perturbations are across a number of different conditions, but then once you've finished all your analysis, are you able to go back and actually figure out which perturbations actually
Yeah,
have
yeah. So we did do that. Yeah, and so and a couple of different perturbations were relevant. So we already knew that methyl jasmonate is the common stress hormone that a lot of people use to sort of broadly activate defense metabolism. That works okay. But the problem with that is that you activate so many other genes that it's not super specific. We also saw that that chitosan, which is a marker of fungal infection, was pretty decent for activating that early pathway. Um, but but again, like we were able to test via metabolomics a smaller subset of the conditions than we could test by transcriptomics, just because it's less scalable. Yeah. So we might have still missed through the transcriptomic approach the absolute ideal condition. Um, yeah.
That's fascinating. Thank you.
I see that
Yeah, we
there's
have.
a talk or the question in the comments.
Yes, please. Yeah, go ahead.
Um, Someone is asking if the module assignment was done with WGCNA, and it wasn't. I think WGCNA uh, is more tailored to, it's been optimized for a different type of data for bulk RNA-seq, and often the outputs of that are gene clusters, or yeah, gene clusters that are very large. Um, I used this process called consensus matrix factorization, which is out of the Sabeti lab at the Broad Institute. I don't know if you can see the reference here in the corner on my slide. If not, you can see it in the paper, or you can, yeah, you can just search matrix factorization Sabeti lab. Maybe I'll link it in the chat here. But it's a great co-expression type factorization tool that we didn't develop, that had already been developed for mammalian single cell data. Yeah.
Yeah.
But yeah, we did a bunch of different types of analyses. We did some standard like bait-based co-expression correlation. We did some global uh, like hierarchical clustering type uh, clustering, just like WGCNA. All of those work okay. This one worked the best in our hand. Yeah.
Right. We have one question thing.
Yes, great presentation. So my question is the pros and cons of using either the bulk RNA-seq versus the single cell RNA-seq. So my understanding from your presentation is that if you're looking for like not specific enzymes, just like untargeted proteins involved in the biosynthesis process, then probably the single cell approach will work better. But I want to hear your opinion on like when to use the bulk versus the single cell RNA-seq technique for solving this
Right.
kind of yeah.
Yeah. I know I went through this a little bit fast, but I think here, I mean, this slide, we're comparing bulk and this new data set directly. And we're just looking at known enzymes in the pathway. So this is not about discovery of new enzymes. I think one of the issues with Taxus is that there's so many relevant enzymes, so many oxidases and acyltransferases that, and they're often expressed in the same tissues. So the bulk RNA-seq is limited in that case. And so I would say like generally that would be where I would recommend using this protocol is when you encounter a situation where you have a large amount of specialized metabolism beyond the pathway that you are studying that is co-expressed in the same place. And you need to differentiate those by trying to perturb them up and down in a different way so that you can see which genes are part of these different pathways. Um, the yeah, that's what I would recommend. I think it's a useful process for finding not just the accessory proteins, but also, you know, the core enzymes of your pathway. Um, and I think it's not that at the beginning of this project, I was intimidated, you know, by doing the single cell approach and developing um, new technology, but it's really not that expensive or challenging. And, you know, really what we were doing here was um, one experiment where we could test about 200 and something conditions, samples. Each of those samples, we got a lot less coverage than we would with bulk RNA-seq, but we could test so many for about the same price as a small set of bulk RNA-seq samples. So probably this data set cost about the same as like 12 RNA-seq samples. So it's really not that yeah, not that fancy and not that expensive. I see
Thank
your
you.
full
Yeah,
glass.
Jörg, please.
Okay, thanks. Thank you. Lars, do I have permission for a question and a comment?
Sure.
Good. Okay, good. So I ask my question first, and then I make the comment, Connor. So the question is, could you just go to this slide where you show this model of photo connecting the terpene synthase and the cytochrome P450?
Oh, the little cartoon there.
Yeah, this little cartoon. Yeah,
Yeah, this is way
perfectly
out of
there.
date. Yeah, we don't think this. Sorry, this cartoon is a little bit out of date. We do think that that photo is directly interacting with
This,
text.
sorry. Oh, one. Sorry. I'm like listening to a meeting at the same time.
Okay, good. So, sorry.
Yes, I think so. Yeah, this model is uh, a gross approximation and is also out of date. I need to replace this cartoon, but we do think that photo is helping to scaffold things on the ER surface, including 5-alpha and other P450s. Its relationship to taxidine synthase, which is in the plastid, we have less confidence in. We're still trying to figure out what that does. It's possible that photo is going in and out of the plastid and somehow being involved in transport. We can measure binding in vitro. That's the main data that we have with its interaction with the terpene synthase. Yeah.
And okay, I still ask then, like, do you think it's really specific for diterpene biosynthesis, or could it be involved in scaffolding and ER arrangements with other biosynthetic systems?
Yeah, so I guess so. This data is not mine, and it's going to be put
Okay,
on
good.
bioarchive
Okay,
soon. But
good.
I'll vaguely say that we've now found that photo seems to help with yield in other terpene pathways.
Okay.
And so that suggests that maybe it does have some sort of impact on the ER or, yeah.
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 Because when you mentioned that it seems to be highly conserved as a single copy gene, the first thing that came to mind to me was that it might function, it might have an original function perhaps in gibberellin biosynthesis as a plant conserved process where there's not much tinkering possible.
Yeah. Definitely could. We think, having looked at the co-expression data for the Arabidopsis homolog, which is uncharacterized,
Yeah.
it looks like it is co-expressed with a couple of enzymes, and so probably involved in some sort of more core terpene, primary metabolism type terpene, like gibberellin or some steroid or something like this.
Okay,
Yeah.
great. So here comes my comment, and I'll make this very, very quick because you alluded at the very beginning to some of the foundational work from the Croto lab who had mapped
Yes.
out at least much of
Yes.
the pathway considering the time, with
Yeah.
the first gene being published 30 years ago. I should say that when I saw your paper, uh, that's just absolutely brilliant work, Connor. Absolutely brilliant. And I took liberty to share this paper at the time. I think already confidentially a draft that you had showed me with Rodney Croteau, who's now long retired. And I just wanted to pass this on directly to you, that he was just so impressed with this work and so happy to see how you have now been in one paper filling so many of the gaps that his lab was struggling with for 20 years or so. So
Oh,
absolutely
that's amazing
fantastic.
to hear.
Yeah.
Yeah.
So thank you. Thank you.
Yeah, we're kind of in awe of his group and how they've managed to figure out so many pieces of this, with so few tools compared to what we have now.
Mm-hmm.
But yeah, and once his lab closed, progress kind of stopped, which was really a testament to how
Yeah.
good they were.
Until you came back onto the scene. Okay, great. So I leave space for others now to ask.
Thank you. First, Laura, we have a question here in the chat, and then Emily. I can just read the question real quick. Does the matrix factorization approach work below expression systems? I vaguely remember you mentioned that you needed high expression for this to work.
So yeah, it probably would work, but I would say single cell approaches tend not to work very well with low expression, and just co-expression analysis suffers when you don't find high expression. But I would recommend, if you're trying to if you're struggling with low expression like we were, to try the experimental strategy that we did. Let me pull up a slide. We actually did a bunch of single cell RNA sequencing at the beginning of this project, hoping that that alone would be sufficient, to reveal the pathway just by eliminating some sort of cell type, that was involved in production of Taxol. And we saw really low really low expression of a bunch of the genes that we knew were important. So this is just data showing 4 of the genes in the pathway and sort of vanilla single cell RNA sequencing of a bunch of tissues. And it wasn't until we added those perturbed cells in that we then started to see high expression. So especially if you're studying stress-related metabolism, I'd recommend the experimental approach more so than the analytical approach here. Yeah.
Thank you. Emily?
Oh, hi. Thank you so much for the talk. The paper was really great and like very nice and fun to read. The question I have was about the intermediates. When you were showing the slides for the intermediates of the pathway, I noticed the peak heights were very low. Do you have like NMR data for those? And if you were able to get NMR for those like really low expressing intermediates, like were there any challenges of like getting that compound, and how did you overcome those?
Yeah, we didn't get NMR until we got to Baccatin. And most of those intermediates, we had a couple of standards that we could use throughout the pathway, but some of the intermediates in the middle there, yeah, there's based on mass and MS2 alone right now. Yeah. But We could probably for the earlier part of the pathway, I think we could do the scale up and do NMR. It's just it takes a lot of time and effort to do that with 40 or so tobacco plants, and we didn't end up doing that until we got later into the pathway. Yeah.
Great, thank you.
Yeah, Tui, please.
Hi, Connor. Excellent presentation. When you were talking about some protection and deprotection or acetylation and deacetylation of the intermediate, I was wondering if it has anything to do maybe with transporting of these intermediates between cell types, or would it be possible for you to go back to your data and see if maybe there's some sort of compartmentalization of the pathway in different cell types, things like that?
Yeah. I think transport is likely, especially after our first module. Let me we so one of the issues that we have right now is there's a little bit of new data that might help us with this, but we really struggled to identify cell types because we don't have a lot of cell type markers for gymnosperms. Um, However, it seems like at least the first module here, which is the first 7 or so steps, is very distinctly expressed compared to the rest of the pathway, and the predicted product that you would get from this is about 4 oxygenations and 3 acetylations. That is a really dominant product that we see throughout the plant, and so our guess is that that's maybe like a hub intermediate that is shipped from one cell type or one tissue to another. And the acetylations, maybe they help stabilize or maybe they help, um, uh, bind transporters. But yeah, now that we can see we're a little less confident separating the second and third module, but that first module is very distinctive and we can see the product of it clearly in the metabolomic data. We can see the product of it induced by some of our elicitors. And so we're guessing that that is probably produced in a separate place than where the later chemistry is done. Yeah.
Great, thank you very much.
Thank you for every question. Is there any more questions? If not, I would like to thank Connor and kind of end the session here.
Thank you, everyone.